Health and Iman. Today's topic is sexual reproduction. Today we're going to discuss about the sexual reproduction in bacterial cell. As we have already discussed about the asexual reproduction, which was binary fission and endosperm formation. And today we're going to discuss about the sexual reproduction. As you know, sexual reproduction is that type of reproduction in which two parents or the two cells involved and they exchange their genetic material, and in this way sexual reproduction takes place. So this is a genetic combination, genetic recombination. The first word which is comes in mind when you read or listen the word sexual, it means the genetic recombination or the genetic reproduction. So it is that type of reproduction in which there is a formation of new cells, formation of new bacterial cell, formation of new genetic recombination by the through the recombination, through the exchange of the genes, through the exchange of the genetic material. After the exchange of the exchange of the genes or the genetic material, a new combination, new recombination takes place, and new genetic material or the new genotypes. We see a new types of the genotypes. New genotypes comes into existence, and in this way the new individual gets new characteristics and recombinant characteristics of the two cells or the two parents which have these participated in this process. So this is known as sexual reproduction. And in case of bacteria, it's the same, that the new genetic material, new genotype is comes to existence by the exchange of the genetic material from two cells and two homologous chromosomes. They exchange their genetic material in the in the form of their DNA, portions of the DNA, small portions of the DNA. They come in contact with each other and exchange their fragments or their particles, and this is known as the primitive type. This is the most primitive type of the sexual reproduction, most primitive type which takes place in the bacterial cell first of all. That's why it is also known as most primitive type of the reproduction. It differs from the eukaryotic cells, eukaryotes reproduction. Why it differs from the eukaryotic reproduction? Because the gametes and the fertilization does not take place in this process. In the sexual reproduction of the bacterial cells, there is no gamete formation and there is no fertilization. While in eukaryotic cell, in eukaryotes, gametes are formed and exchange of the gametes or combination of the gametes takes place and the fertilization takes place. So this is the difference between the bacterial sexual reproduction and it's bacterial reproduction and this eukaryotic reproduction. So this is the difference between bacterial reproduction and eukaryotic reproduction. So this is a most important difference between the both types of the sexual reproduction, because sexual reproduction just started in the bacterial cell at the first time, and it is the primitive type. Now let's have a look on the different types and how the first thing is that how the bacterial recombinations or the bacterial reproduction take place in the sexual form, in sexual style, in sexual style of reproduction. No fusion of the cell. There will be no fusion of the cell in the bacterial type of the sexual reproduction. As we know that in eukaryotic cell the fusion of two cells takes place, but in this case there will be no fusion of the bacterial cells. Just the DNA DNA portion, small portion or small fragments of the DNA is donated. Donor donate these fragments and recipient just receive these fragments. There is no fusion of the two types of the cells. And what happens in the recipient cell? In the recipient cell, donor DNA, it gets recombine, or it get in the recipient cell alongside the recipient DNA. Their fragments comes inside the cell and DNA fragments from the donor cell, and it become join alongside the recipient cell alongside the genes, and homologous genes are comes in front of each other. They are adjacent to each other. In this way the enzymes produces the nicks. As you know, nicks are the small breakdowns in the DNA fragments, DNA molecules. So this is known as the nicks. Enzymes cut out the fragments and produces the nicks, and in this nicks replacement of the fragments or from the donor DNA to the recipient DNA. The fragment of the donor DNA get replaced with the fragment of the recipient DNA. The portion of the donor DNA get integrated into the recipient DNA, and the part of the recipient DNA is replaced with the donor DNA, and in this way the chromosome or the DNA is now becomes the recombinant. Why recombinant? As you know that because it has now the both. So it is the recombinant. Recipient DNA becomes recombinant DNA because it has the portions of both the donor DNA as well as the recipient DNA. Here are the methods of the bacterial recombination. We have studied about the recombination in the bacterial cell, how it takes place. Here is the methods. There are three methods for the recombination or the sexual reproduction in the bacterial cell. The one is the transformation, second is the conjugation, the third one is the transduction. Okay, here is I just sort out some main points and definitions, which is the exact to the to the point to learn about these three types, and then in the next episodes we're gonna discuss in detail one by one all of these. Okay, the first one is the transformation. As its name shows, that transformation is the transfer of the cell-free DNA from one cell to the other. Just the cell-free DNA which we're gonna transfer from the just to cell-free DNA from the one cell, it's gonna transfer into the other cell. Just from it is because it is cell-free DNA, so one DNA we're gonna transfer it into the other DNA and it's gonna change its characteristics. So this is the transformation. Next is the conjugation. Conjugation is that process in which transfer of the genes between the two cells, but they come in physical contact with each other. So this is the point, which is the physical contact. You see here this point, that they come in physical contact with each other. So for the conjugation, two cells, they come in physical contact with each other for the transfer of their genetic material, as they produces conjugation tubes or all these processes we're going to discuss in detail in next videos. And transduction is the third type, and it is a unique type of sexual reproduction in which transfer of the genetic material or the DNA or the gene from one cell to other by bacteriophage. This is the point, that bacteriophage transduction takes place from the bacteriophage. It happens like this. A bacteriophage, it gets the DNA from the one bacterial cell, and then it transfers this DNA from bacteriophage to the another bacterial cell. So in this way transduction takes place, and the characteristics of the recipient cell becomes recombinant and change. So this is the methods of different types of the sexual reproduction in the bacterial cells. So we're gonna discuss about all of these types in detail in coming episodes. So please stay tuned, and thank you so much for coming on my channel. Don't forget to like, share, subscribe and comment, and don't forget to follow me on Instagram. Bye bye. Allah Hafiz.